everyone, and welcome to Mindset Learn Extra. My name is Permi, and yes, I am happy and excited to be with you guys again today as we are going over life science with Aslam. Hello, Aslam. How are you?
How are you doing, Pumi?
I'm good, thanks. And you?
I'm excited for this. Is the last
paper.
Life Science Grade 12 Live 2013.
Oh, wow. I'm sure the matriculants at home are also excited, at the same time a bit nervous.
Nervous, yeah.
Yeah, hey. But hey, guys, I'm sure that you all know that you always have to keep calm and relax and write everything that you know and that you have learned, because right here on Mindset, we are really willing to help you with everything that you are doing. Anyway, guys, right next to me, I have this nice Samsung gadget. In order for you to win, make sure that you jump right there on our Facebook page. It is www.facebook.com/win/learn. You can actually get to go there on Facebook and see what you need to do in order for you to get to win yourself. We're already giving away 4 of the Samsung gadgets, and we have 2 more drawers to give away. So make sure that you keep your eyes locked to our Facebook page and right here on the show. Right now though, guys, let's get learning more and learning extra. Aslam, take it away.
Thanks, Pumi. What an introduction. Welcome, guys, to the last show, 2013 Life Science Grade 12 Live. You must be counting the hours now, because just some hours away at 9 a.m. tomorrow, you're going to be opening that paper and seeing all the questions that are coming there. If you've done well in paper 1, at least you've got 1 step in the door already. If you haven't done so well, all is not lost, because if you cover up in this paper, then your average mark can still go up. So nothing is lost yet, guys. So keep working hard. And remember, no need for tension, no need for nerves now, because proper planning prevents poor performance.
Absolutely.
And if you've done that, then you are ready for the paper. However, let's just remind ourselves again. I always like to do this because I still find learners, up to last night on my WhatsApp, a learner asked me, so what are the topics in paper 2? God, come on, guys, if you are coming to the end of the year and you're asking me what are the topics in paper 2, that's not on. You're supposed to know what's in paper 2. And we've been saying it on this show and everywhere else. We've been saying, this is paper 1, this is paper 2, this is paper 1, this is paper 2. But let's revise that anyway. Paper 2, we're only looking at paper 2 now. We're not worried about the other paper. It's gone, done and dusted. So paper 2, we start with plant hormones, or what I prefer to call plant growth substances, but nonetheless the textbooks call them that. And then we go to animal responses to the environment, and that is broken up into the central nervous system, receptors, and obviously peripheral nervous system there, and also chemical coordination, also known as endocrine system. We then move on from there, animal responses, we've done that. Then we look at also thermoregulation in humans. Only in humans is prescribed, by the way, in your exam guideline. No other animals, no other examples are prescribed. If they do ask you questions on that, they would have to give you enough information in the question. Thermoregulation, and then we go into reproduction. Reproduction is broken up into reproductive strategies. These are just terminology and ideas how organisms ensure that reproduction takes place. Then we move into plant reproduction, and then we look at human reproduction. In this whole thing, we must also know the difference between asexual and sexual reproduction and the advantages and disadvantages of each. And finally, then we move into the environmental topic, and this is broken up into population studies, community, and social. And that, people, is your paper 2. So need to know that. And you'll find, you follow us here now while we're doing the questions. We've taken questions from almost each of these topics to help you along your way. So let's get the show on the road. Let's kick off with a question on plant hormones. adapted from 7th September 2013. That's this year's trial exams, KZN, paper 2, question 2.2. The following diagram shows the results of an investigation into the growth responses of roots and shoots to auxins at different concentrations. So the topic obviously then we're looking at is plant hormones, and we're talking about auxins and what it does to roots and shoots at different concentrations. So here we have our graph there showing you the concentration of the plant hormone in milligrams per molecule per mole there, and inhibits and stimulates. There's your zero there. Above that would be stimulating growth, and below that would be inhibiting growth. So let's look at what we're reading here. As you can see here, from about already just 0,00 there, we're getting some stimulation of growth there, and in high concentration there's an inhibition there in the root. Whereas the stems are not stimulated or inhibited in the early, once it reaches 0,4 it starts stimulating, and when auxins concentrations are high it stimulates growth, and when it gets too high then it starts inhibiting growth eventually there. So first and foremost your homework is to read the graph. At what concentration of auxins do stems show the most growth? Obvious, here we will look at the stems. So we're not looking at root here, we go to the graph, we look at stems. Where's the highest point? That's the point there, and they made it easy for you. There's a line going down, and your answer is 0,6 there. And let's check that. 0,6 milligram per mole. And from what concentration of auxins is growth inhibited in roots? So we go to the roots, we go to the line, below the line is inhibited, above the line is stimulated. So we look at this, here's the point in that area there, sorry about that, in this area there. We're going to take our ruler from that point onwards there and go down, sorry again about that. Take our line and go down there, and our line is going to be between 0,3 and 0,4. Remember, I am not using a ruler, you're going to use a ruler for that. And the story is 0,2 there. Let's see, can't be 0,2. It's still not being inhibited. Let's look at the question again. From what concentration of auxin is growth inhibited? Growth inhibited in group. Look at what the question is saying, inhibited. Let's go back to the graph. All of this, above this line, it is still being stimulated. So the answer there is incorrect. It should be then from above, just over 0,3 and 0, you can say 0,3, 1, 2, 3, 4, 5, whatever, not 5 because it would be in the middle. Between 0,1 and 0,3 or 4 would be the 0,3, sorry, 0, where's my pen? 0,3, 3 to 4. It would be more or less more accurate, but they would give you, obviously, because there's no units there, they would give you some leniency there. Which part of the plant, the root or the stem, is more sensitive to auxins? This is a nice question, and this question is important in understanding also something more important, which I'll outline just now. So, which of these two organs is stimulated or is affected with a lower concentration of auxins? Obviously, the root, as you can see here. So, if the one that is stimulated at a lower concentration, that means it needs just a little auxins to make a difference. So it is more sensitive. You know, if I pinch you, and if I pinch softly and you jump, that means you're sensitive to the pinch. And if I pinch you and nothing happens to you, Pumi, that means you're resistant. Yeah. And when I press harder and harder, then we can measure that it takes you so much longer than somebody else to feel the pain, isn't it?
Yeah, so, true.
Or if I and Pumi and I decide we want to know how sweet something is, or which one of us tastes something that is equally concentrated in sugar, which one of us feel that it's sweeter? And I take one spoon, she says, nah, this is not sweet, and I say it's very sweet. Mm-hmm. So that means I am more sensitive to sugar. So that's how you understand this concept, guys. This is how you understand that. It's easy, just that you need to just read more into the graph than meets the eye. Now also look at what's happening here. What happens at a higher concentration of auxins here, and this is not high compared to high there. You can see already that the root growth is inhibited there, and in the shoot growth is stimulated up to very high concentrations of oxygen, then only it would come down there. So you must keep that in mind when we're talking about phototropism and geotropism. In this case, obviously, our answer would be the root. Suggests how the information in the graph can be used to manufacture more effective weed killers. That's easy. Roots are sensitive to low oxygen concentrations. A weed killer will only lead a low concentration of oxygens to kill off the roots, and if you kill off the roots, the weed will die. There's no sense just killing the shoot, because then the root will survive, and the root will then grow more shoots there from there. So we don't need much there. Explain the effect that oxygens have on stems when exposed to unilateral light. A theoretical question, straightforward answer. First of all, we say that the light from one side will cause the oxygens to move to the shaded side. They move to the other side, the non-illuminated side, the shaded side, the dark side, whatever you say there. Increase in oxygen concentration there stimulates cell division and cell elongation. So this side will grow faster than the unshaded side, causing the shoot to bend towards the light source, and the phenomenon is known that we say that the shoot is positively phototropic. Notice that we have simple sentences there. One is when a plant is exposed to light from one side, the oxygens move to the other side. Number one. Number two, the oxygens, from the functions of oxygens now, stimulate cell division and cell elongation. In other words, on this side, because there's more oxygens, the cells are going to divide faster, they're going to elongate faster. And what does this mean? This means that this side will grow faster than the other side. Which side we're talking about? The side that is not receiving light. What is that? The dark side, the shaded side, the non-illuminated side, whatever you want to call it. But it's the side that does not get light. So what happens? That side is now lopsided in terms of growth compared to the other side, so therefore the stem will bend towards the light. Again, remembering the elbow, there's more skin on that side, there's less skin on this side, therefore you are allowed to bend your elbow this way. You cannot bend it the other way, otherwise the bones will break here, number one. Number two, the skin will break because there's too little skin to allow bending in the opposite direction. Apart from the fact that the bones are also, it's a hinge joint, doesn't allow movement in it. But even if it could, and if the skin structure was the same, then you wouldn't be able to bend in the other direction because you have too little skin on this side. Huh? Or if you're rowing a boat, remember, if you want to turn right, you row more on the left. If you want to turn left, you row more on the right. So think of these things when you're talking about oxygens. So this one here is talking about phototropism and the shoots. We can also take this and we say that if, for example, we lay a plant flat, let's go right down and open up some pages here. Okay, let's say we take a plant and we lay it flat like this, horizontally. Now, if this is the root end and this is the shoot end, what's going to happen? Because of gravity, remember here there's no light. We're just leaving the plant without any light. Light from all over, all around, number one, or we can put it in a box to negate the effect of light. What can happen here is all the oxygens will come here at the bottom. Because of gravity, under the influence will push there, not below there, by the way, where I'm showing it, on that root and stem, but at the bottom end. I'm just showing you the area there. So in other words, it would be, if I can get this right, around that area there. I'm doing great here. Around that area there. So you're going to get a lot of oxygens there. Now what happens is, in a root, oxygen inhibits growth. In a shoot, high stimulates growth. So here the stimulation will take place here, and therefore the shoot will grow upwards because this side is going to grow faster than the top side. And in this side here, this side, the inhibition is taking place, so it's going to grow slower, so therefore the root is going to go downwards. We say that the root is positively geotropic under gravity, and the stem is negatively geotropic. Remember, because we're not talking about light here, we can't talk about phototropic. Again, let's distinguish between those terms. Tropism would be a response to external stimuli. Notice it's very general. Nothing, we're not defining what that external stimuli is. When we add photo in front, then we're talking about light. When we add geo in front, then we're talking about gravity. So this is what, when you're defining terms also, remember, if they don't say response to light. Oops, my fingers, I'm a magician, you see, I just touch the screen and everything starts moving. We just say response to external stimulus, it will be tropism. Response to light stimulus, photo. Response to gravity, geo. Chemical, chemo. Water, hydro, etc. So guys, that is that section. In addition to that, you must know the functions of gibberellins. You must know the functions of abscisic acid and oxons. And in addition to that, you must know how hormones are used in agriculture. The advantages of using hormones in agriculture. If you have questions on that, you can post it on the page. Thank you. Pumi.
Oh yes, guys, we are still going the distance with you. I can see a lot of you are asking for the race number. It will be posted on our Facebook page. Yes, I can see. Oh, okay, cool. It is there on our Facebook page. So make sure that you jump on there right there at www.facebook.com/learnextra/win. It is just as simple as that. Right now, guys, let's take a break. We'll see you shortly after this. Welcome back from the break, Mindsetters. We are still going the distance with you guys at home. I'm sure that you are tuned in and ready to go our next segment with us as we are revising life science. As you know that matriculants are writing tomorrow their life science paper 2. Let me just say a quick shout out to Puleng from Leribe and Lesotho who is watching the show, and of course Kwezi Selekeleni. I'm sure that you guys are also going to be asking all of your questions that you have, because tomorrow you guys are writing at 9 a.m. So make sure that you bring them all and then we'll try and answer them as far as possible. Right now though, let's keep on learning. Aslam?
Thanks for me. Remember, guys, if we don't get to your questions, it's not a train smash. It's not a train smash.
Yeah.
We would be able to help you via the help desk as well. So those
yes.
questions are forwarded to the help desk, and we can then try and help you via email, etc. And on the page as well. Later, we would try and answer some of them. So don't stress if we don't get to your questions, but we want your questions there so we can clear up some of the stuff. So in terms of what we've spoken here, we've done plant hormones. Let's move on then to another question, and we start with the nervous system. And the nervous system, remember, in the nervous system you got to learn the brain and the 4 things in the brain that you need to know, 4 parts of the brain that you need to know: cerebrum, cerebellum, hypothalamus, medulla oblongata. Know them on the diagram and know their functions. Remember, in exams, not always do they just ask you straightforward the functions. They can ask you what happens if these things are damaged, and that's the reverse. That means if this is doing that, that won't take place, etc., etc., etc. And you must also know the difference between the central nervous system and the peripheral nervous system, and you must know the autonomic nervous system, and there the sympathetic and the parasympathetic. Only focus on the responses of the sympathetic. Remember, the sympathetic nervous system and adrenaline are very similar in function, and the parasympathetic nervous system takes the body back to a state of relaxing. The sympathetic takes it to an excited state, and the parasympathetic takes it back to relaxing. So you need to know the opposite reactions there in terms of the eye, the heart, the blood pressure, the whole lot of other stuff that happens there. I'm not going to go into details there. That's what you have to learn. We're looking at a question. I found something different on in the September 2013 KZN paper again. And let's see what they say. Study diagram below, which shows some of the structures involved in a reflex action. So you already know that they're talking of a reflex action when a girl picks up a hot plate and answer the questions that follow. So this young girl decided to go and touch a hot plate, and something happened. As you can see what's happening to her hand, she pulls it away, and we can talk about that. Define the term reflex action. That's easy, guys. Straightforward question. It's a rapid automatic response to a stimulus. They're using involuntary there, same as automatic response. All right, you must be able to say this involuntary. You are not controlling it. Identify the parts labeled A, B, and D. A would be the muscle. In this case here, you don't need to know the name of the muscle, or you could say the effector, because that's what it does in this section. That's A. And then B. B is showing the receptors in the finger. You must try and, you can say finger there, and then, but we're not talking about a body part, so you're not in grade 3. We're in matric now. We are looking at what in this reflex action or reflex arc, what is that representing? The receptor in the finger. Come on. All right, and the last one was D, spinal cord. That one is straightforward. Okay, all right, we open that. Straightforward labeling, there we go. All right. Explain one advantage of a reflex action. That's very easy. Prevents a person from causing injury or harm to themselves. I would still go further to say from causing, let's get the pen going again, causing further. Remember, once you carry this hot plate, you've already got injured in somehow, you got burnt. All right, maybe not injured, but if you didn't have a reflex action and you didn't put it away, you would have got burnt further or worse in that way. So we're saying prevents further injury from harming the body. Explain the effect of the body on the body if part labeled C was damaged. Part labeled C, it's a neuron here. Remember, diagram not very clear, scan, and the arrows are showing going up that way towards the central nervous system. So we need to then look at that. C is a sensory neuron. So first you do is you name the neuron. Remember, sensory neuron is also called afferent neurons in some textbooks. If you're having a problem remembering which one is afferent, which one is efferent, rather stick to sensory neuron. That carries a stimulus from the receptor to the CNS. Stimulus will be received and will be converted to a nerve impulse by the receptor, but the nerve impulse will not reach the spinal cord. It will not reach the spinal cord. Huh? Okay? It will not reach the spinal cord. And there will be no appropriate reaction because now the message is not getting to the spinal cord, so how are you going to respond? So you can't respond, although you have the ability to respond because there's nothing wrong with the motor neuron, but you're not getting the stimulus in the impulse from the stimulus in the first place. So you can't let go because you don't even know that it's hot. What is the role of part E in the reflex action? Part E is referring to the brain here, and we want to know what does part E do in this reflex action? After the reflex action, that means after you drop this pain, after you've dropped the plate, not the pain, after you drop the pain, something's wrong with the tongue. The brain and the tongue are not in coherence today. Something is happening. Something's getting lost here. The reflex action of the cerebrum registers the sensation of pain. In other words, what happens is the pain sensation only goes to the cerebrum thereafter. You're burning, you're picking it up, and you're dropping it off. So the part of the brain that's doing that, it obviously, because the brain, cerebrum, interprets and receives and interprets all sensations. Pain is a sensation, is a feeling. So therefore, it would be there as well. So that's what it does. It doesn't really take part in a reflex action, but it registers the pain afterwards. Okay, good. So that's the reflex. Remember, they can also give it a reflex action in various other ways. They can give it a reflex action as an essay. They've done it in the past already. You have seen that question where they ask, do not, when they ask in an essay, write down the reflex action as a flow diagram. As, for example, you write stimulus, a receptor, impulse, and you write there, motor neuron, response. This is a flow diagram. It's not acceptable in an essay. So you do not write it like that. Remember, I said last week, it seems so long ago, I said on a Thursday that you can write in point form. This is not in point form. This is a flow diagram. You must say something. It must make sense. The stimulus is received by the receptor. It's converted to an impulse, etc., etc., etc. I'm not going to go into too much detail there. You must be getting the point. Okay. Now here is a big passage, and we're not going to go through this whole question. Adapted from September 2013. Are you posting the questions on the page?
Yes, they are.
Yeah, okay.
They are.
So you're getting this question. You can go through it on your own. I just want, it's a lot of reading here. You're going to take too much of time. I'm exposing you to this question. They're giving you a story about Alzheimer's, and then they're asking you questions on Alzheimer's. So I'm not going to go into the whole question. You need to know that even in the brain, you need to, over and above what I told you, you must also learn the disorders, that is depression, Alzheimer's, ADD, and then you must also learn a little bit about the general effects of drugs and alcohol on the nervous system. You don't have to go into too much detail. The general effects of, and the very common ones are, it slows your reaction, it can affect memory, it brings about moods and aggression, etc. Across the board, paranoia, they all have more or less those things there. So generally what drugs and alcohol will do to the brain and spinal cord, the nervous system in general, you need to know that. So I'm not going to do that question. Let's move on. And remember, that's an example of a comprehension question where you would have to read and give answers from here and from your knowledge. They're going to ask you questions on the theory, and they're going to ask you questions from here where the article is saying that when they ask you from the article, you must quote from the article. Okay? That's the important thing, is the exposure that you're getting. Okay, then we look at, now we move on from the nervous system, and the central nervous, peripheral nervous system, we now move to receptors. And we stop at the eye first. Free State to question 2, 2013. Study the diagram and answer the questions that follow. It's a diagram of the eye, as you can see. They give you a caption there. Write down the letters only of the part which. Okay, number one, level 1 of the eye, you've got to learn all the parts, not just the parts that we're asking here, but you must learn all the parts, number 1. You must know the functions of the different parts. You must know how we see. You must know what happens when something is near, something is far, accommodation. You must know what happens when we're seeing in the bright light or in dim, that is pupil mechanism. And you can go further into structural suitabilities of the eye. Let's go through this one. Write down the letter or letters only of the parts which undergo a change when the intensity of light changes. Now, they gave you 2 marks here. Take a guess, how many letters will there be? Obvious, there must be 2. They're not going to give you 2 marks for 1 letter. So there must be 2 letters there. Light intensity changes will affect C, the iris, and B, the pupil. They only ask you for the letters. Sometimes the question says, provide the letters and the names. Remember, if you do one of the 2 only, you're going to lose half the marks. So in this case, they only ask for the letters. So it would be B and C. Our answer here would be B and C. 1 mark, 1 mark. Next question says, undergo a change when the distance of the object from the eye changes. And that means accommodation. Remember, when we're talking about light, it's the pupil. When we're talking about near and far, it's the lens and associated structures. Get that straight in your head. When we're talking about light, you can't talk about near and far. When we're talking about near and far, you can't talk about light. They're not related in the sense. Okay, so which are and how many marks? 3. So obviously we're looking for 3 structures. And there would be obviously H, what is attached to H, G, and what is G attached to D. It would be D, G, and H. D, G, and H. D being the ciliary muscle and body, G being the suspensory ligaments, and H being the lens itself. Okay, may lead to total blindness in one eye when it is damaged, and that would be E, the optic nerve. Total blindness if the optic nerve is damaged. If any of the other parts are damaged somehow, you would be able to see in that eye, but you'll be seeing less, you know, unless of course we take away the whole retina, but the retina is not labeled here in this case here, so we can't even talk about that one. So the most likely answer there would be E. Okay, we can argue about that one there. Lots of people can argue about that, but for our studies, let's just keep it there. Why are you being funny with me now? Why are you being funny? Okay, there we go. And is rich in blood vessels which supply the cells of the eye with nutrients and oxygen. That would be the choroid. We look in the diagram and choroid is F. So our answer would be F. That's a basic question on the eye. More for section A. Sometimes they can ask you in section B. They can then expand this into near and far and bright and dim. Now look at this question on the eye. This is slightly more difficult. Study the graph which shows changes in the shape of the eye shape of the lens over time. So it's telling you least convex here and most convex. That means here it's flatter, here it's rounder in simple terms for us. Okay, time in seconds going that way, and they're telling you G, F, E, and D. For number 1 says name the process which is represented by the graph. What did they say? Changes of the shape, most convex, and they're talking of the lens, and I told you earlier when the lens is being spoken about, it would be accommodation. Which letter represents part of the eye when reading a book? When we're reading a book, that's near vision. The eye must be as convex as possible. So we go to the diagram. That's convex there, most convex. Go across, and the letter is D. Okay? The letter is D. Which letter represents the eye looking at an object moving towards the viewer? This is an interesting question. Moving towards a viewer. Now let's look at this diagram or the graph. There we have G, we have F, we have E, we have D. It can't be D because we already said that that's reading the book. Now it can be any one of these. Now let's look at these letters and let's look at here. Here no real change has taken place. It's in the same spot all the time. Here what has happened? From being less convex, it's moving towards being more convex. Here in this area, from more convex, it's moving towards less convex. And here, from E to D, it's moving from less convex to more convex. But look at this letter here, F. F is by this whole line. The others are at specific points. So the answer is F. If they had another letter here, it would have been interesting to see what learners would have done. Okay, F, G, H, we call that H. Lots of you would have chosen that one, but it can't be. It's this one here because it's least convex moving towards more convex there. Okay, and that would be that one there. F, describe the changes. We'll take this one after the break.
Oh, yes, guys. Aslam, we have this one, did you know, question coming from one of the mindsetters, but I wanted to confirm it right after the break. It is coming from Matsubane Minitefo. It says, did you know that humans see light wavelengths from 390 to 750 nm? Humans hear frequencies of 15 to 20,000 hertz. So we are going to be confirming that right after the break, right? Interesting. Okay. All right, then, guys, let us take a quick break, and when we come back, we are still going to be learning more and learning extra. Stay tuned. Welcome back, Mindsetters. We are still going the distance with you, and yes, we are doing life science revision. Great also writing life science paper 2 tomorrow, so I'm sure that you guys are all relaxed, having a glass of water and eat a lot of orange veggies and fruit, right?
There you go, and nuts.
It does help. It does
Brain
help.
food, protein.
Yes. Before the ad break, we had that did you know question. Yeah. So what is the answer to it?
No, it's just a fact, yeah.
It's just a fact.
Those are just interesting facts. Just remember, mindsetters, you don't have to learn that. You cannot be examined on that. Huh? Those are just interesting things to know. Anything else we got that's important?
We also have a mindsetter asking here, it's Tondani, asking, will it affect my sight if the ciliary muscle are damaged?
Obviously, if the ciliary muscles are damaged, then they're not going to be able to contract and relax properly, and that will affect the state of the suspensory ligaments, and that will be affecting your ability to alter the shape of the eye. So obviously that would be there. Is that it?
Nope. There's one more from Nemutu Tanyi. Nemza asking, are you allowed in an essay to write in a tabular form comparing two things?
What's her name?
It's Nemza.
Nemza, come through the camera here so I can smack you one hard one. Remember, I told you last week, and I told you earlier as well. You cannot tabulate. You cannot give a flow diagram. You must write an essay. You answer the essay. You don't have to put it in a paragraph fancy like a composition or something, but you must be able to write it down. No tables, no flow charts, no diagrams. Is that it?
Yep, it is.
Okay, there we go. I think we've exhausted that question now. Just remember that. Guys, remember, if you have other questions, please post them. The last question on the eye was, describe the changes that occur in the eye which result in a change in the shape of the lens from point E to point D. Lovely question. From point E to point D. Now notice what's happening. It was flatter, least convex, to more convex. So we are moving from far to near. So we have to describe near vision here. And very easy that. The ciliary muscles, not circular, let's take that out. Just talk that ciliary muscles contract, the sclera is pulled forward. Lots of books are also saying that it's the choroid that's pulled forward. Let's debate around that, so you could say any one of that. But notice what's important is point this one. This point is very important. The suspensory ligament becomes slack. Notice these two are antagonistic. If that contracts, the suspensory ligaments become slack. If that relaxes, the suspensory ligaments become taut, T-A-U-T. Elastic lens bulges and becomes more convex. Light rays are reflected more. The clear image falls on the retina. Before that, we can also put here the focal length decreases. And the clear image. Remember, when you want to see whether the thing is far or whether it's close to you, the image must always fall on the retina. Let's do a little bit of logic now. If the image must fall on the retina and the thing is close to you, then obviously you don't need a long focal length, you need a shorter focal length. And if you need a shorter focal length, then the ray must be refracted more, because refracted means bent. The more bending takes place, the shorter the focal length. How can we achieve that if we have a convex lens? How can we get a convex lens if the suspensory ligaments are not pulling the lens apart, so they must be slack? How can we achieve that if the ciliary muscles contract towards the lens? Then we have this. Can you see the logic going backwards? We don't have to explain it that way. Now what happens if we're saying in the opposite of this? It would be the opposite answer. Let's see how we can change that just here without changing this whole thing here. If we want to see something far, then I'm going to write there far, so if you see this later, you're not going to get confused. If you want to see far, then the ciliary muscles will relax away from the lens. And the sclera will go back to its normal position. And the suspensory ligaments become taut. They do not contract because they are ligaments. They do not have the ability to contract. Only muscles have the ability to contract. So we say they become taut. And elastic lens bulges. Now we say the lens becomes flatter. And when this happens, less refraction. All right, you're saying refracted less. And the focal length now increases. And can we change the last statement? No, we can't change the last statement, because whether we see near or far, the image must fall on the retina. So can you see how easy that is? Don't learn both, learn one, because the other one is the opposite. The other question they can ask you here is for dim and bright. Remember, you need to know that in dim light, the pupil becomes wider or it dilates, so more light enters. And for that to happen, to actually happen, let's just extend the page there. For the pupil to become wider or dilated, it's in dim light. I use this here for you to remember. DRCR, in dim light, the radial muscles contract. And we put a square there. The circular muscles relax. Of what? Radial and circular of what? Where are they found? On the iris. And the pupil dilates. More light enters the eye. And again, if you want to change that, I use BCCR squared, I think, if I remember, yeah. Okay, here now, in bright light, the circular muscles will contract, CC, and the radial muscles will relax. The pupil becomes constricted, and less light enters the eye. But we don't even have to remember that, because we can do it here. Relax, contract, constrict, and less. That's all that you need to know there. Okay? Very good. Let's move on. We'll come back to that if somebody has a question later. Study the diagram below and answer the questions that follow. Oh, sorry, we haven't answered all the questions on the eye. We're just jumping the gun here. For me, what's happening to me today?
And it's not a lot of questions, though.
Okay. Name the disorder of the eye that tends to affect people older people, and people suffering from diabetes. This would be cataracts. Okay. How can the disorder mentioned in question 6.2? Something missing. Be treated, obviously, would be the answer. Be treated. This would be treated. Be treated. Surgical procedure to remove them, or laser treatment to reduce clouding on the eye. What they do is they make a small incision on the eye, they remove the lens and they replace the lens, suction out and pushing back the reverse of the suction, putting another lens in, so the clouding of the eye is removed in that way. Good. I think that's all I didn't answer there. Let's move on to the ear then. Remember, also on the eye, you must know questions on near vision, not near vision, sorry. Somebody is short-sighted and somebody is long-sighted. Remember that. We'll come to that a bit later as well, if we have time. Study the diagram below and answer the questions that follow. Identify parts A, E, and F. A is the pinna. E would be the, it's not that part, it's this part here, that will be the cochlea. And F, this part here, would be the tympanic membrane. Good. Got it all. Explain how the function of the ear would be affected if the bones of part B were fused. The bones, the ossicles, hammer, anvil, stirrup. Remember that. To remember the individual names, HAS, has, hammer, anvil, stirrup. Their purpose, what is their purpose? Their purpose is to take the vibrations from the eardrum and onto the oval window. But they don't only do that, they amplify the sound, they make it louder. Why? Because we're coming from the pinna, which is a large surface area. From there, the tympanic membrane, which is smaller than the pinna, but it's still large compared to the hammer, anvil, and stirrup, and the oval window. So we're going from a large surface area, we're taking the same amount of sound waves and we're concentrating it on a small surface area of the oval window, and therefore we intensify or amplify the sound in that way. So what's going to happen if we fuse those bones? And that would be taken from the function. The sound vibrations would not pass through the inner ear, would not pass effectively. You can't say cannot pass through the inner ear. They would still pass through the inner ear effectively. And hearing would be affected. Notice they don't say that you become deaf. You can't become deaf because there will still be vibrations, but they will be less. And you will be, obviously we can talk about that the sound would not be amplified. Okay? Write down only the letter of the part, which is concerned with equilibrium and balance. Notice you must not confuse equilibrium and balance with balance of pressure in the ear. Okay? Which part is concerned with equilibrium and balance? In this diagram, it would be C and D. C and D, equilibrium and balance. These two parts, the semicircular canals and the vestibule, which is made up of the sacculus and the utriculus, both of them play a part of in balance and equilibrium. So it would be those two there. What will you say? C and D. Where the impulses for hearing are generated, impulses where they are generated. Now somebody would say, hey, man, impulse and auditory nerve, how come we're not going there? Notice the question does not say where the impulses are carried, where they are generated. Where is an impulse generated? At the receptor. Where is the receptor in the ear? In E. A pupil asked me today in my class at 10 o'clock this morning, he asked me, how are the retina of the eye and the cochlea of the ear related? Nice question. I found that to be interesting from a pupil, and obvious, they are both receptors. The cochlea has the organ of Corti, which converts sound waves into impulses, and the retina has the rods and cones, which converts visual images into impulses. So they are both receptors within their respective structures. Okay, so the cochlea is our answer there, and that would be E in this case. Notice in the ear, you need to also know how the ear is suitable for its functions. The large pinna, which can trap sound waves, the grooves on the pinna that direct sound waves in, the fact that the pinna is outside the body, trap sound waves, the funnel shape of the pinna. Then we go into the auditory canal. It is hollow and long to direct sound waves. It also has hair in it to trap and remove dust particles. It also has mucus or wax inside to do the same. The eardrum is a very thin, tight membrane, so any disturbance in sound waves is going to cause it to vibrate, and its larger surface area compared to the oval window helps to amplify sound. The three bones, their size, etc., and their shape allow them to amplify the sound. The oval window, these semicircular canals, they are posed in different directions, as you can see, 1, 2, 3, so they can detect movement in any direction, movement and speed, any way you want to move. You can use that for that explanation there. Good. Inside the cochlea, we have the fluid which assists in the organ of Corti as well to do that. Remember, you do not have to know too much detail of the cochlea and the structure of the organ of Corti and so on, but you need to be able to say how we hear, and that is that sound is trapped in the pinna, goes into the auditory canal, causes the tympanic membrane to vibrate, that causes hammer, anvil, stirrup to vibrate, oval window vibrates. All of this causes the fluid in the inner ear to vibrate. This causes the organ of Corti to vibrate, and when the hair cells touch the tectoral membrane in the organ of Corti, the hearing sound waves and vibrations are converted to an impulse auditory nerve, from the auditory nerve to the brain, where it is interpreted. That would be on the ear. Good. Also on the ear, you need to know that the semicircular canals and the sacculus and the utriculus, they help us in balance. How? Semicircular canals, as I told you now, they have cupula, semicircular canals. The C sound is predominant. Semicircular canals have cupula. These cupula inform the body about the direction and the speed in which somebody is moving. They pick it up that way. And the utriculus and the sacculus, they have macula. Sacculus and utriculus have macula, some. To remember that, the sum. Sacculus and utricula have macula. The macula has jelly, has hair, but it has these little marble-like structures, otoliths, which serve as balance. They are, in terms, sensitive to gravity. And they will tell us about the position of the head compared to the rest of the body in terms of balance and equilibrium. So therefore, when you're going to the merry-go-round and you're going round and round and round in different directions, your brain is getting information from all these guys in different directions. At the same time, your food is also moving up. You know, you may have some reflux, and this is what causes nausea and dizziness when you have these long rides. With that, I think we'll take a break. Yep. After the break, we've got something interesting coming up.
Yes, we do indeed have something very interesting coming up right after the break. But before that, let me say a quick shout out to Kaloshia Desiree, who says, hey, can you please tell Aslam, thanks so much for all the help during their exams, made life science easier. Thanks once again. And of course, there is also Sahal Hello saying, shout out to all the Nazruddin learners watching this. I want to thank Uncle Aslam for teaching so awesomely. All the best. And of course, there's one more shout out to Sfiso Ndlazi from Esogazi Secondary School. I'm hoping that you really are going to be making yourself and the nation proud. To all of the matriculants, all the best, but we'll see you shortly after the break. Welcome back, Mindsetters. We love all of your questions. Over the break, we went over some of the questions that you asked, and there is Keegan Canterbury, Mr. Aslam, who asks, will we need to know how the ear and balance work in full detail for tomorrow's exam?
We obviously have to know how the ear works in terms of hearing, but like I told you, the way I explained it just now, that's all you need to know in terms of hearing and in balance. That's all you need to know. Don't go into too much detail there. You need to know what the semicircular canals do, what the sacculus and utriculus do, and how we hear from the outer ear right up to middle ear and in the inner ear. Just a brief little story about the vibrations that are transferred into the cochlea and how the stimulus is converted to an impulse and how it goes to the auditory nerve to the brain. That's it. Don't go into too much detail about the structure of bony labyrinth and membranous labyrinth and so on. Look at your exam guideline. Those things are not prescribed.
Okay, thank you. And there's one more coming from Christopher Lee, asking us to please go back to question 2. Would it not? Oh, hi Aslam. Would it not be 0.2?
Yes, that's a turning point.
Yeah, because it's a turning point.
Okay, Christopher. What's your name, Christopher? Eh?
Yeah, it's Christopher.
Okay, Christopher. Let's look at the question again. This is 0. Anything above this line, according to this graph, is stimulating. Anything below is inhibiting. Let's go back to the question. The question says, where's the question? Where's the question? All right, there we go. From 2. What concentration of oxygen is growth inhibited? Inhibited. So can it be 0.2? Let's go back to the diagram. 0.2 is that point there. Christopher says that's a turning point, so therefore it starts inhibiting at that point. But all of this, where is it? It's above the 0, yes? And anything above the 0 is stimulates. Yes, the level of stimulation is less as we're going down there, but there's no inhibition. Inhibition means it stops the growth. Here there is growth taking place, but the effect is less. Below the line, that is where my argument is. But we can debate about this till the cows come home. Some people will see it differently. But mine is in the English language. They ask for inhibit. Inhibit in this graph is below 0. So only at that point does it start inhibiting it. Up to the point that there's even one dot above this line means there is some stimulation of the growth in that particular route, but it's not as much as it is there. So keep that in mind when you're answering that question. Good? Any more questions there for me? Or shall we hold on to the question?
Yeah, let's hold on.
Okay, let's move on to another question from KZN. The diagram 1 below shows the position of certain endocrine glands in the human body, and diagrams 2 and 3 show the effects of endocrine control in response to external stimuli. Study the diagrams and answer the questions which follow. I like the diagrams. They give you one diagram on one side that has all the glands, and here they're telling you something. There's a snake there, and it's hissing, and this girl is in a state of shock, and this guy, he's training in the hot sun, and they're telling you he is sweating. Okay? Identify the endocrine glands labeled A and C. That would be A would be the one there, the base of the brain, and that would be the pituitary gland, and C on this side is the thyroid gland. Okay? Let's see. There we go. Hypophysis is another name for it. Write the letter of the gland from diagram 1, which causes the girl in diagram 2 to react in a manner shown in the diagram. That means she is in fright mode. Flight or fright, in this case here. But it's not flight or fright, it's flight or fight. Fight or flight. You either stand and fight or you fly away. In other words, you run away from there. In this case, she's in fright, she's in shock. Okay? So which gland here helps her to be in that state? And that would be number D, letter D there, obviously, the adrenal gland. Okay? Name the hormone which is secreted from this gland. That would be adrenaline. And list 3 effects of the hormone named in question 2 that enables her. First of all, it increases her heartbeat. And if it increases the heartbeat, you must remember the moment you talk about adrenaline and you say it increases the heartbeat, the next thing you must also talk about is that the blood pressure increases. Now I'm talking about in a general question. Because if the heart is a pump and it's pumping faster, then obviously the pressure in the blood vessels will be higher. So blood pressure increases there. It increases the tone of the skeletal muscles in her case now. That's important. It also increases the depth and rate and depth of breathing. Why? Because she needs to take in more oxygen and release more carbon dioxide so she can supply the cells, these skeletal muscles, with fresh oxygen so that they can metabolize quickly and get enough energy. Constriction of blood vessels to the skin and digestive system. Why? Because the digestive system and the skin are not vital organs in an emergency. And at the same time, dilate the blood vessels and divert this blood to the heart, voluntary muscles, and the brain. Because these are considered to be vital organs in an emergency, so more blood can go to them so that we can think clearly, quickly, and we can use the muscles effectively, and obviously the heart can pump blood properly to all the organs. Name the hormone secreted in large quantities by gland A by the person in diagram 3. What's happening to that guy? He is sweating. So that means he has lots of water, and therefore the hormone that involved water would be ADH, antidiuretic hormone. You just say ADH is good enough. The hormone identified in question 83A will be produced in smaller quantities when the person in diagram 3 stops exercising and drinks some water. Name the mechanism which causes this to happen. That would be negative feedback, because here now he'll have more water, so therefore he'll need less ADH. Remember, when it comes to negative feedback, guys, you need to know that negative feedback simply means negative feedback is a mechanism where a homeostatic, we can say it's a homeostatic mechanism where a stimulus results in an opposite, or rather in a response in the opposite direction. You'll notice when you're talking about that, if it says was an increase with the stimulus, the response was decrease. Too much will become less. Too little will become more, etc. This is what a negative feedback mechanism is. And the main example that you study is with pituitary gland and the thyroid gland. When there's too much thyroxine in the body, then the pituitary gland releases less thyroid stimulating hormone, which in turn will stimulate the thyroid gland less, and therefore the thyroid gland will secrete less thyroxine and the level will come back to normal, and vice versa in the opposite direction. Now, it's not only that gland that works that way. Here in this ADH, we can talk about the same. We can talk about it in glucose and in insulin and glucagon, etc. We can use it in most of our endocrine glands. So you must be able to respond that way. We move on, guys, to the question on thermoregulation. And here it's a straightforward question. Study the diagram which represents the human skin and answer the questions that follow. Name the part in the brain that regulates body temperature. These are straightforward questions, so we're just going to move on. Hypothalamus, obviously. Provide labels for parts marked A and B. A and B would be the epidermis and the dermis. And notice in the diagram of the skin, you must concentrate on those parts that are involved in thermoregulation. And the study guide tells you, the exam guideline tells you that in thermoregulation, you must only worry about the role of the blood vessels and the sweat glands. A little bit about the muscles and shivering in the cold, but generally you must worry about the sweat and the blood vessels in the skin when you're doing that. So those are the labels you must worry. Remember, your textbooks and your knowledge will tell you about erector muscles and the hair. Scientists are saying that the hair in humans doesn't play such an effective role. Therefore, it has been downplayed, and therefore, you do not get credited when you talk about that. You can include thyroxine also on a cold day. You'll get marks for that because it's part of our work. Which diagram, 1 or 2, represent the skin on a hot day? You've got to look at the blood vessels, and you've got to look at the sweat. There and there, there and there. That's all you need to know in thermoregulation, so you've got to look at that. In this diagram, the blood vessels are constricted. Here they are dilated. Notice I'm not saying thicker and thinner and all that. I'm saying constricted and dilated. That's the words you must use. Here there's less sweat. Here there's more sweat. These dots represent sweat. So obviously, from your own knowledge, you know that wherever there's more sweat, that will be the diagram that will show on a hot day, and that will be diagram 2. And in this diagram, just by looking at these diagrams, we can explain. Use these diagrams. You've got it on the page. Use these diagrams to explain what happens on a cold day, what happens on a hot day. You can just take one. For example, on a cold day, the hypothalamus sends messages to the blood vessels of the skin to constrict. And if it constricts, less blood goes to the surface of the skin. And if less blood goes there, less heat goes there. And if less heat goes there, less heat will be lost by conduction, convection, radiation. By the way, you don't have to know all of that, but less heat is lost there. Okay? And then that way, that means we're keeping the heat in the body. In the same token, the sweat glands are less active, and they secrete less. They don't say they secrete no sweat, because sweating takes place all the time. It's just that on a colder day, there's less sweat that will be secreted. And therefore, if less sweat is evaporated, there'll be less cooling in the body. If it is very cold, then the brain sends messages to the muscles, the voluntary muscles, and they start contracting and relaxing faster, and we start saying that we are shivering. Shivering causes friction in the muscle, and obviously the metabolic rate will increase and the body heat increases. Or we can bring in thyroxine here and say thyroxine increases, this increases the metabolic rate, and therefore more body heat is formed. I'm not going to go on a hot day, you can work that one out. I've given you one, you can work out the other one. Give 2 reasons why that one is there. We've already discussed that, about more sweat and blood vessels are wider or they are dilated. Name the condition that results when the core body temperature of a human drops below 37 degrees for a long period of time. That would obviously be hypothermia. Hypo, less, lower temperatures. Hyper, lot, hyperactive. If someone's hyperactive, it means it's too active. So hyperthermia is when the temperature remains above 39 degrees for a long period of time. List 2 symptoms of that would be dizziness, decreased metabolic rate, drop in heart and respiratory rate, numb feeling in the hands, even you can be getting blue on the ends there. Also, you can talk about the person that is delirious, he doesn't know what he's saying, he's confused, etc. in that way. Good. That brings us to the end of thermoregulation. Remember, in a few years ago, or rather last year or the year before, they asked you for thermoregulation in the essay, where they linked the adrenal glands and the hypothalamus and how they affect the blood vessels. The adrenal glands causes the blood vessels of the skin, I'm talking about, to constrict when in an emergency. And the hypothalamus causes the blood vessels to constrict on a cold day and dilate on a hot day. So you would have had to explain hot day, cold day, and the functions of adrenaline in an emergency. So you can get an essay there. In the eye also you can get essay, near vision, far vision, they can put it together, and pupillary mechanism, they can put all that together. Okay, so there's quite a bit that you can get in all the sections that we have done. Even in auxins, explain phototropism, geotropism, and explain the role of auxins or other hormones in agriculture. So every section that we have done so far can be, an essay can come from any section in those ways. The idea is the essay is testing whether you know your work. They want 17 facts, give the 17 facts. We move on to reproduction, your favorite section. Adapted from there, we start with plants first, study the schematic representation of alternation of generation in a plant, and answer the questions that follow. They say identify the generations labeled A and B. Now, when you want to know the generations, there are only 2, sporophyte and gametophyte. Gametophyte, the gametes will be found in that generation. So in this diagram, where are the gametes found? They're found in this direction here. They're coming from B. So B is the gametophyte generation. A will be the sporophyte generation. Another clue is haploid. Gametophyte generation is generally haploid. So all of this is haploid. Above here will be diploid, because you can see these 2 are going to fuse just now. But let's answer the questions. Identify the generations labeled A and B. We've done that already. Which processes are represented by the boxes number 1, 2, and 3? 1, 2, and 3, meiosis, 1, mitosis, 2, and fertilization, number 3. Okay, those are the 3. Provide the chromosome condition of the structures, number 2 and 4. They can also ask you provide the ploidy. Another word there would be provide the ploidy. In other words, what is the condition of the chromosome number? Is it haploid or diploid? And your answer there would be number 2 is haploid, because it's coming from haploid there, all the way there. And number 3 is the result, n plus n gives you 2n, which would be diploid. So that's how they ask you this question on plant life cycles. You need to know this one. I can ask you, of which plant is this? Is this the moss or the angiosperm? Your answer would have to be the moss, because they're talking about spores. In the angiosperm, they're not talking about spores, okay? So you need to know that as well. You must be able to look at the flowering plant or the angiosperm, its life cycle diagram. In addition to that, you must know the structure of the flower. You must know what pollination is, cross-pollination, self-pollination, wind-pollinated flowers, and insect or bird-pollinated flowers, and the differences between them. You need to know that as well. And you need to know reproductive strategies, that is oviparous, ovoviviparous, viviparous, altricial, precocial, internal fertilization, external fertilization, courtship behavior, etc. Just how these enhance reproduction in an organism. With that, Pumi.
Oh yes, the mindset is Aslam are feeling so confident about tomorrow's paper. I'm sure that you guys are really relaxed and learning so much, because even I am learning so much from all of you guys posting and answering each other's questions on the page, which of course we confirm to see if you guys are answering well the other mindsetters. And again, let me just say a few shout outs to those mindsetters that seem confident. It's Bilal Adam, Ipeleng Bridget Mudise, Angel Tusi, just to name a few. And shortly after the break, I will try and ask some of the questions that you guys have asked us right here on the show. We'll see you shortly after this. Welcome back from the break. If you just joined us, we are still doing a life science. We're going over revision with for life science for tomorrow's paper. Make sure that you stay tuned with us, because we have other interesting questions coming from the mindsetters at home wanting to know. It is Priyan Govinda. They, Aslam, in the T. Isaac's guide, they highlight that...
Can I summarize that question quickly?
Oh, you want? Okay,
yeah.
cool.
Right, we want to, it's a long question. Sorry for me for that. The learner wants to know that in one textbook they say that the hypothalamus secretes ADH, and the other textbook says it's pituitary gland. They're both correct, but there's a slight difference. The hypothalamus actually, in the guideline, look at your exam guideline, the hypothalamus secretes ADH. It's not the pituitary gland. Hypothalamus secretes it. It is stored in the pituitary gland, and it is released when necessary from the pituitary gland. So you need to know that the ADH is secreted from the hypothalamus and not the pituitary. Okay, that was the one.
Yeah, and another question coming from Maureen asking, may you please explain how pancreas acts as an endocrine and exocrine gland?
Excellent question. How is the pancreas an endocrine gland and an exocrine gland? Remember, the pancreas from nutrition grade 11 secretes pancreatic juice. When it does that, it secretes that pancreatic juice into the pancreatic duct, and then it goes to the intestine. So because it's secreting it in a duct, we say that it is an exocrine gland. An exocrine gland will secrete in a duct, and from the duct it goes to wherever it needs to go. And an endocrine gland, on the other hand, is a ductless gland. It secretes its secretion directly into the blood. And where in the pancreas this happens? When insulin and glucagon are secreted, they are hormones. Remember, all hormones, that's why we call it the endocrine system. All hormones are secreted directly into the blood, not into ducts and tubes. They go straight into the blood, and the blood is the transport system. Good.
Can I please take one more? It's Mpiwe Mkhuno. How is the stimulus turned into an impulse in the ear?
How is the stimulus turned into an impulse in the ear? Remember all the vibrations up to the oval window, then it gets into the cochlea, and when it's in the cochlea, the vibrations continue. The organ of Corti, and you don't need to know the structure, I'm explaining it just for your understanding, is made up of hair cells and a membrane above the hair cells. When that hair cells touch the membrane, the stimulus is converted to an impulse. Okay, I can move on.
Yes, you can.
Good, guys. Now we're moving on to reproduction. The two graphs show the changes in temperature in a woman's body, as well as the level of hormones, estrogen and progesterone during the menstrual cycle. Typical question here, showing you what's happening here. Changes in the level of estrogen and progesterone in the woman's body at different temperatures. This one here, sorry. Is dealing with that one. The one on top is the temperature. Notice from day 0 there right up to day 12, 13, 14, temperature is relatively stable, drops a bit, and then it goes up. It stays above. And look at we're looking at a very short 0.5 degree rise, and then it comes down again after day 28. And in terms of the levels of estrogen and progesterone, we see estrogen going up there and staying relatively high and then coming down. progesterone rising after estrogen later much and then coming down. Between which days does menstruation takes place in this woman? Again, this is a straightforward question. Anything from 28 to 7 or 0 to 7 also you can say there. Calculate by how many degrees Celsius the woman's temperature varied in one menstrual cycle. You have to show the working. I'm not going to go into the whole details. You're going to take the highest, you're going to take the lowest, and you will then work it out that way there. 12.3, from the graph, list 3 pieces of evidence which indicate that ovulation has occurred, and that would be the temperature increased, progesterone level begin to rise, and the estrogen level decrease after ovulation. Just after ovulation, you'll see there's a slight dip in the estrogen level as well. Good? Explain the importance of the higher level of progesterone from day 15 onwards. Again, what are they asking you? What is the function of progesterone? And that is progesterone to maintain the thickness of the endometrium, uterus lining, and to prepare the uterine lining for the embryo. And we say that progesterone maintains pregnancy. So should fertilization occur and pregnancy take place thereafter, obviously, then progesterone levels will be high. Describe the relationship between the levels of progesterone and the changes in the temperature in the graph. That's straightforward. You're going to read from the graph. I'm not going back up there. The levels of progesterone increase as the temperature increase, and decrease as the temperature decrease. You'll read that from the graph. Good? So that's a graph question. Sometimes they give you this type of question also to identify the different hormones. Remember what I told you when we did this section. Remember this word FOLP. FOLP. FSH first, then estrogen, then LH, and then progesterone. So when you're looking at a graph that gives you these different hormones and how they're thickening the endometrium, look in the middle, you'll find LH. That's the one. The middle day, that means in the cycle, if it's 28 days around the 14, the highest hormone would be LH. Anything after that, that rises after that is progesterone. Anything that rises before that is FSH and estrogen. FSH has to rise before estrogen because follicle stimulating hormone comes from the pituitary gland, which develops the follicles. The follicles thereafter, when they're developing, secretes estrogen. So you know that order when you're answering a question on a graph. We were going to do the essays later, but we've pulled it up further. Now you can see what question number it is. 23. It was the last question for today, but we've brought it up because we see the need to bring that earlier. From puberty to menopause, KZN, by the way, a woman's body undergoes a complex series of cycles. Describe the role of all the female hormones in the control of the menstrual cycle, and include in your essay how the cycle changes with pregnancy. What do you have to do? You've got to explain the 2 cycles. That is the uterine cycle and the ovarian cycle, with specific references to the role of the hormones. That means the FSH, estrogen, LH, and progesterone. Right? You need to know that. Then after that, you'd say, if pregnancy occurs, what's going to happen? The levels of progesterone remain high to maintain pregnancy. FSH is suppressed in this time, and so on. So you're going to explain exactly what I told you there about the FOLP. If you know that, you can answer this essay. The other essays go around the same thing about negative feedback between FSH and progesterone. Again, you need to know the hormones, their functions, and where they come from. When you're saying any hormone, FSH, secreted by the pituitary gland, and it does X, Y, and Z. In this case, not X, Y, and Z, but it stimulates the development of the follicle. O, estrogen, secreted by the follicle. What does it do? It prepares the lining of the endometrium. L, LH, secreted by the pituitary gland. What does it do? It stimulates the Graafian follicle to release the egg, ovulation. What happens then? The Graafian follicle becomes the corpus luteum and secretes progesterone, which does what? It further thickens the endometrial lining for possible pregnancy. Later, we get prolactin and oxytocin. Prolactin to stimulate the breast to develop and to secrete milk, and oxytocin when it's time to give birth to stimulate the muscles of the abdomen to contract and relax. Good. Then I'm not going to discuss this question. What I'm telling you here is a question like this would ask you questions related to these structures. Remember, we're seeing here the placenta, we're seeing the chorion, And in some questions, you may even see the amniotic fluid. And they will ask you functions on the amniotic fluid. They will ask you questions on the placenta. They can ask you questions on the umbilical cord as well. And in terms of the umbilical cord, remember you have an umbilical artery, an umbilical vein. Umbilical artery takes blood from the fetus to the placenta. Therefore, it will be filled with waste. And the umbilical vein will take from the placenta to the baby or the fetus, and therefore it will be filled with useful substances like food, oxygen, and the rest of it, opposite to our body. I'm not going through this question, like I said. Also remember, you must be able to know what happens at birth. The waters break, and the muscles start contracting and relaxing to push the baby out. Thereafter, the doctor will remove the afterbirth, and so on. That you need to know. That's just general. We're moving to the environment now. I'm going to skip that one there, ecological footprint. You can do that. Look at this essay. Discuss the role that carrying capacity, competition, predation play in regulating the size of a population. Carrying capacity is the maximum number of individuals a particular environment will hold. That's the first thing. And with it comes the environmental resistance. How? When organisms reach carrying capacity, the environmental resistance will come in because there's only so much of space, etc., etc. And this will keep the population below the carrying capacity. Competition, obviously, only those that are fit will survive, and that will keep the population down as well. Last one, predation. Remember, a predator and a prey. In this relationship, the predator keeps the prey population in check, and the prey population keeps the predator population in check. You need to explain how this relationship, which one I'm talking about, the predator-prey relationship, the one that goes like that. Right? When the prey population rises, the predator population also rises. But as it rises, the prey population decreases. So in this way, they're keeping each other's population constant. If we remove any one of them, it's going to have an effect on the other one. So that's the other type of essay that you can get there. Okay, and in this one here, just want to show you quickly, not that one. Mark recapture you can work out, it's very easy. We just want to move on to a graph question I'm looking for. This is the one. Is it this one? Yes. Right, this graph will show you logistic growth. It's not an S graph, it's not an S growth, it's logistic. It has these different areas. The lag phase, the log or accelerating phase, the decelerating phase, and the equilibrium phase. Guys, if you need to know those phases and explain what is happening in each of those phases to know what is happening there. They also gave an essay, Gauteng 2012, last year, they asked the question, explain the two growth forms. The more marks will come from this one because it has more. And sometimes the graph may even come down, have the death phase. You must be able to say what is the carrying capacity, what is environmental resistance, etc. when you do a graph like this. And you need to talk about everything that's happening there. Carrying capacity, just again, the total number of individuals that an environment can support. Guys, I want to say thank you to all of you. We wish you all well, whatever you're doing. My family at home, I hope you do well in terms of you've given me time to come and do this, so the whole country benefited because of you. I tell you all that, and one particular person, Yasmin, has been watching me and spreading the word in KZN for us. Thank you very much. Pumi.
Oh yes, thank you so much, Aslam, and the mindsetters at home. Thank you so much for watching the show. This is actually the last live show for Life Science 2013, and it is. Okay, cool. Okay, cool, guys.
Mindsetters, I leave you with this.
Mindsetters want to see this. Whoa. Great.
There you go, mindsetters. I'll leave you with that famous trademark of mine. Thank you very much.
Bye-bye.